Hello everyone, this is solving past paper 3-1 summer 2014. Now in this paper actually the wet lab is divided into two parts. The first part aims to study the effect of different temperature on the time needed for Benedict to change in color. So the first change in color will be recorded in the table versus the temperature. And in order to do that, simply we need to do the same concentration of glucose and the same volume of glucose, same volume of Benedict. The only thing we change is the temperature, and simply we can do that by changing the temperature of the water bath. So what you're going to do is to get a test tube, you're going to add 4 centimeter cubic of glucose, and you can add the same volume of Benedict. So that helps you in deciding the volume of Benedict that you need to add. You add the same volume, so the ratio of 1:1. You place it in the water bath at one of those different temperatures. Now, in order to obtain the temperature, you need to heat the water bath, and then you start measuring the time using the stopwatch. Now, let's see, for example, how our students test for the time or measure the time needed for Benedict to change in color at what temperature is that? 90. So let's try. So would you please? Yeah. So they are placing the test tubes with Benedict at 90 degrees Celsius, and then they measure the time until the first appearance of color or the change in color. And as it is stated in the question paper, it should be at the top. Now, I think it's 90, and you need to turn off the Bunsen burner because it's going to be more than 90 if you can. Now, here's the change in color. What was the time?
23 seconds.
After you do that, simply you place it in the table of results, which is temperature per degree Celsius and the time per second. And after you do that, you can find out which one is the fastest change in color. So those results right here on the board are one of our students' results. It shows that as the temperature increases, the time needed for the first appearance of color decreases, and the time is basically per second. Okay. Now the second part of the experiment is basically to find out the concentration of unknown glucose solution. Now, in the question paper, there is S1 and there is S2, and both of them are unknown. So in order to find out the concentration of those unknown solution, we need to make a standardized test. And as you know, guys, in order to do a standardized test, we need to make a range of concentration. Now, in this question paper, the range of concentration can be made by the serial dilution. Now, the serial dilution, as it shows in the question paper itself, this serial dilution reduces the concentration into half at every concentration. So the concentration of G is 4%. Now, the second concentration is 2. Then you need to conclude that the third would be 1, and the fourth is 0.5%. In order to do so, we need to take equal volume of both the G and the water. So 15 centimeter cubic was taken from the G, and then 15 centimeter cubic is taken of water in order to make this concentration, and that results with 30 centimeter cubic of 2%. Now you can repeat it again. You can take 15, and then you add 15 water, and then you're going to take another 15, and you add another 15 of water to take this concentration. After you do so, after you make those different concentrations, you need to take a specific volume of each one of those and to add Benedict on them and to place them in water bath at same temperature in order to measure the time needed for the color to change. Now, before doing that, let's see how to make the dilution in practice. So the dilution simply can be made in practice by taking 15 centimeter cubic of G, and then we can add 15 centimeter cubic of water. So here is the G, which is the original solution, the stock solution. So 15 centimeter cubic of G using the syringe. That's 5. Is that 5? So simply you need to fill the syringe 3 times. So this is 1, So again, here's the G. You're gonna take, that's 10 centimeter cubic syringe, okay? Then that's 15. So she used the first syringe with 5 and then she used another syringe with 10. You know what? I recommend to use the same exact syringe, okay? And then she's gonna add 15 centimeter cubic of water. Okay, now you need to shake it to make sure that it is homogeneous concentration. Now that solution, the new one is 2% glucose. Did you label the beaker? Did you label the beaker? Yes. Now you need to label the beaker first. Is it labeled?
Yes.
Can I? Yes. This is one. Is that the 4% or the 2%?
4%. Here is the 2%.
Now this one, it should be this 2%. Why is that? Because you get 15 of the 4. Now the G is the 4%. So once you take 15 out of the G and you take 15 out of water, you're gonna make 2. So this one should be labeled by 2, not 4. Okay? This is a mistake, but we can learn out of our mistakes, right? Simply you can erase the labeling. So scratch on that labeling, scratch on it, and then that's gonna be 2%. And the other beaker should be 1%. Great. And then it's gonna be 0.5. Great. Now in order to do the 1%, what you're gonna do? You're gonna use, wait a minute. Now this is 2%, right? This is the 2%. So you're gonna take 15 centimeter cubic of the 2% and you're gonna add it into this empty beaker, which is labeled by 1, and then
we're gonna add, we'll take
15 and you're gonna add it into the 1. Excellent. And after that you do the same with the 0.5%. You're gonna take from the 1 and you then, yes. Okay, so you add water. So you have 2 syringes for water and 2 syringes for the solutions? Come on, you are cheating. You're not supposed to do that. You have only specific number, limited number of syringes in the exam. Okay? You're doing smart. So you are working smart, not hard. Okay. Anyway, the idea is if you are using the same syringes, you need to wash them out. Okay, so you are making the last and the final concentration. So we are learning many things out of there. Yeah. Okay, those simply how we do the dilutions. Now after we are done with the dilutions, simply we need to test. Using Benedict, we need to test the time needed for each one of those different concentrations, the time needed to color change at a specific temperature. So as we hear, simply we take specific volume. How much you took? 5 centimeter cubic. So you take specific volume of each one of those concentration and place it in the test tubes. So those test tubes are basically labeled. Okay. Organization is very important. What's that? This is 0.5, and this is the S1. So you're gonna do it for S1 and S2 at the same time. Great, which is the unknown. So concentrations, same volume, and now you're gonna add Benedict. Add Benedict into the test tubes. How much you're gonna add? 5 centimeter cubic. Okay. In each one of them. So let's see how you do that. So you're gonna take 5 centimeter cubic and you add at every one of those. Great. So this one is ready now to place in the water bath and for the time to be measured. So what temperature are you gonna use?
80.
You're gonna use 80 degrees Celsius. Why 80? Why it's not 70? Why it's not?
It was easier to maintain than 70 and...
It was easier to obtain. It was easier to obtain. Great. Thank you very much. So carry on. So for example, here is the 4%. The 4% glucose concentration at what temperature? 80. It's supposed to be 80, but as you can see, it drops. The temperature drops. And that's basically one of the sources of error in this experiment. The temperature keeps changing. Anyway, we need to carry on. We can improve this experiment by simply placing insulators in the water bath, or we can use thermostatic water bath. So we're gonna measure the time until the color. Is there any change in color? No change till now. So we're gonna measure the time needed for the first color to change, and we're gonna repeat for all the concentrations: the 2, 1, 0.5, S1 and S2. Okay? And if we do that, we are doing basically the standardized test. Is there any change? It's gonna take some time because it dropped. Okay. I think there is a color change. Stop your stopwatch, and that's 2 minutes, which is 120 seconds. 120 seconds, and here is the first color change. Okay. So we're gonna take your results and put them on the board. Okay, then those results was taken by one of the students at temperature 80. So he decided to take temperature 80 because it was easy to obtain. At 0.5, 160, 142, 120, at 4, 100 seconds. Now, for S1 and S2, S1 it took 140 seconds, S2 it took 104. Now, if you estimate the concentration of the glucose in S1 and S2, S1 is going to be somewhere between 0.5 and 1, okay, because it took 140 seconds. And for S2, it's going to be somehow above the 4, okay, somehow above the 4, because it took 1.04. And those basically was one of the students' results at temperature 80, as I said. Now we need to mention the sources of error again. There are two main sources of error right here. The first one is misjudging color, and the second one is changing temperature. Now, in order to improve, guys, you need to use thermostatic water bath, and the second one you can place white paper behind, and you can, for more accurate estimation, you can simply use a smaller concentration of glucose. Okay, thank you for watching and have a nice day.